FISH, Principles and Methodology. Fluorescent in situ hybridization or FISH is a technique in which single-stranded nucleic acids, usually DNA, sometimes RNA, may also be used. They are permitted to interact so that hybrids are formed by molecules with sufficiently similar complementary sequences. Through nucleic acid hybridization, the degree of sequence identity can be determined and specific sequences can be detected and located on a given chromosome. The method comprises of three basic steps: fixation of a specimen on a microscope slide, hybridization of label probe to homologous fragments of genomic DNA, and enzymatic detection of the tagged target hybrids. Probes can vary in length from a few base pairs for synthetic oligonucleotides to larger than 1 mega base pair. Probes of different types can be used to detect distinct DNA types. PCR amplified repeated DNA sequences, oligonucleotides specific for repeat elements, and cloned repeat elements can be used to detect clusters of repetitive DNA in heterochromatin blocks or centromeric regions of individual chromosomes. Chromosome painting with fluorescence in situ hybridization or FISH. Cytogenetics entered the molecular era with the introduction of in situ hybridization, the procedure to locate the positions of specific DNA sequences on chromosomes. Since the first in situ hybridization experiments in 1969, many variations of the procedure have been developed and its sensitivity has increased enormously. Fluorescence, a phenomenon whereby a chemical excited at one light wavelength emits light at a different and usually longer wavelength, is used throughout the life sciences to study a wide variety of structures and intracellular activities. Today, most in situ hybridization procedures use fluorescent probes to detect DNA sequences, and the process is commonly referred to as FISH or fluorescence in situ hybridization. The success of FISH and all other methods of in situ hybridization depend on the remarkable stability of the DNA double helix. In molecular hybridization, a labeled DNA or RNA sequence is used as a probe to identify or quantify the naturally occurring counterpart of the sequence in a biological sample. In 1960s, researchers Joseph Gall and Mary Love Pardue realized that the molecular hybridization could be used to identify the position of DNA sequences in situ, that is, in their natural positions within a chromosome. Preparation and hybridization process RNA. Cells or frozen tissue sections are fixed and permeabilized to allow target accessibility. FISH has also been successfully done on unfixed cells. A target-specific probe composed of 20 oligonucleotide pairs hybridizes to the target RNAs. Separate but compatible signal amplification systems enable the multiplex assay, up to 2 targets per assay. The signal amplification is achieved via a series of sequential hybridization steps. At the end of the assay, the tissue samples are visualized under a fluorescence microscope. For example, RNA fluorescence in situ hybridization for Cre mRNA shows genetically identical cells in which expression of the CRE or Cre is epigenetically regulated. KAP104 is a control gene whose expression is not epigenetically regulated. Preparation and hybridization process DNA. First, a probe is constructed and tagged directly with the fluorophores, with targets for antibodies or with biotin by nick translation or PCR using tagged nucleotides. Then, an interphase or metaphase chromosome is firmly attached to a substrate, usually a glass. Repetitive DNA sequences must be blocked by adding short fragments of DNA to the sample. The probe is then applied to the chromosome DNA and incubated for approximately 12 hours while hybridizing. Several wash steps remove all unhybridized or partially hybridized probes. The results are then visualized and quantified using a microscope that is capable of exciting the dye and recording images. Fish testing is a method by which an assessment is made for the presence, absence, relative positioning or the copy number of specific DNA segments by fluorescence microscopy. Depending upon the application, fish can be applied to metaphase chromosome preparations or interphase cell nuclei. Metaphase fish studies or microdeletion fish. Metaphase fish studies are designed to detect changes, that means the microdeletions or microduplications associated with specific phenotypic findings, and require specific and focused clinical questions to be addressed via appropriate DNA probe selection. Genetic changes that are detected in metaphase chromosome preparations by this type of testing are limited to position and copy number changes, primarily losses or deletions, and in some instances gains, that means the duplications of the specific chromosomal regions for which the employed DNA probes are localized. Repositioning of these DNA probes from their normal sites must be determined in metaphase cell preparations, that is translocations, insertions, etc. However, these types of study do not rule out other forms of genetic abnormalities, which may include low level of tissue specific mosaicism and or other forms of molecular alterations like single base pair mutations, uniparental disomies, etc. Interphase fish studies. Enumeration or rearrangements involving specific DNA probes are the only information available from interphase fish studies. Additional inferences of the chromosome constitution of cells utilized are not possible from interphase fish studies. Aneuploidy screening in interphase fish studies does not address structural content of the chromosome detected. The use of prenatal interphase fish studies as an in vitro diagnostic test has been cleared by the FDA only as an adjunct test to standard cytogenetic analysis.